what I want to do now is just finish up the parts of the notes we're going over today are sort of just additional information. It's just interesting to talk about. It's not really on your quiz tomorrow. So let's talk a little bit about how genetic technology has started to allow humans to do things that have been much more difficult to do in previous times. So first off, there's this term biotechnology. It's basically using modern technology to be able to alter the genes of an organism. Now, humans have been trying to influence the genes of organisms for tens of thousands of years. And the most common example of that is in agriculture. In agriculture, humans, once they started to grow crops and raise animals, they started to look at how they can change the characteristics of the crops and of their animals. Like, so where things that we use for crops or livestock or pets, where did dogs come from? The dogs, the ancestor of modern dogs are wolves. And humans selectively chose which wolves they would allow to breed and they would allow to live. And over a period of thousands of generations, wolves started to change, okay, into what we call today dogs. And that process is called selective breeding, choosing which organisms thrive. We talked about this when we talked about evolution. Crops arose from lots of different wild plants, okay, that humans selectively bred, choosing the ones that tasted the best, grew the largest, were disease resistant, etc. And that allowed humans to sort of guide how these species evolved. But today, we have a much more advanced way of doing this. Today, we have what's called gene splicing. Now, this is not what it actually looks like, because DNA is a very, very small, microscopic molecule. Scientists don't use scissors and glue and tape to mix genes from two organisms. They use chemicals. They use enzymes. And so what humans have now been able to do, scientists can take a gene that's in the DNA of one species, cut it out, and insert it into the DNA of another species. And so the DNA of that species it was put into now can start to express whatever trait scientists inserted into it. And so I'll give you a good example of one of the first successful areas where this was done was in making human insulin. So you probably know people that are diabetic, and they have to give themselves insulin periodically because their pancreas is not making insulin. So they have to give it. They used to do it mostly through injections. Nowadays there's insulin pumps and patches and lots of other ways, but it's still just insulin. So 50 years ago, people that were diabetic, their insulin came from pigs. When pigs were slaughtered for food, their pancreas was removed and some of the insulin extracted, and that was given to people to use. And it worked. Pig insulin worked pretty well in humans, helped people to control their blood sugar. However, it didn't work perfectly. Some people were allergic. It was very hard to get. It was expensive. So to solve those problems, what scientists did is they found, in human DNA, they got the instructions about how to make insulin. Instructions in our DNA that tell our body how to make the protein insulin. They cut it out of human DNA. Then they took some bacteria and they inserted this insulin gene into the bacteria and put it back in there. And then they allowed those bacteria to reproduce. But these bacteria are special. They have some human DNA in them. So as they grow, that's what they do.
They turn to human.
They don't turn to humans. They make insulin. Their DNA has the instructions to make insulin, and they make it. And then so people can grow them in these huge vats, and these specially engineered bacteria produce all of this human. It's identical to human insulin because it came from our DNA. And so they extract the DNA from these bacterial cultures, and that's where today insulin comes from that people take for to help manage blood sugar. So they can cut and paste these genes from one organism to another. That's one example. But there's lots of other examples. So there is they took a gene from a spider that makes spider silk, insert it into a goat, and the goats produce this protein that goes into sort of the material of spider webs, and they produce it in their milk. And then people can extract it and use that for different purposes. Like in bulletproof jackets, this material is very strong, and it can be used there. They can take genes to make certain vaccines and put them into vegetables. So rather than going to the doctor for a shot, you might just eat a potato that has the materials of the vaccine sort of built into it. Corn. They produce corn plants that have natural pesticides that they produce in their leaves that when you grow these corn plants, insects won't eat it. Okay? So it helps grow corn more efficiently. Okay? This cabbage, scientists placed a gene from a scorpion into it that produces this chemical that kills caterpillars. Okay? And so caterpillars will no longer prey upon it, but this doesn't affect humans. So injecting DNA to produce a virus in a banana. These are all examples of genetically modifying organisms. And so the hope is also that at some point we could go in and fix genes. So many diseases are genetic in nature. They're because of chain mutations in our DNA. And so scientists hope that we could maybe someday, and we're close to being able to do this, go into and replace defective genes in a person's body with corrective genes that help solve or cure certain illnesses. Sickle cell disease is caused by one mutation. One of those G's, A's, T's, and C's is incorrect, and it causes a person's blood cells to be misshapen, and it causes a whole host of health problems, and can cause early death and severely painful bouts when your blood clogs arteries and capillaries. And so if they can go in and just change this one letter in a person's DNA, then they can help to cure that disease. Certain types of cancer are genetic. There's a BRCA gene that puts a person at much elevated risk of breast cancer. And so you can be tested for this gene. Like I did 23andMe, you ever hear that? So, and it gave me a list of all these things. And one of the things it reports on are health issues that are found in your DNA. So my cells, I don't have that BRCA1 gene, which is good for me and for my daughters and so forth. But if a person is found to have it, and doctors can go in and change the gene into a corrected version, then it might be able to drastically reduce certain types of cancer and other diseases. So there's a lot of opportunities, and for you guys as you go on to high school and college and professions, if you go into the medical field, like genetic medicine is a hugely growing area of medicine because our tools and the technology related to advancing is advancing at a rapid pace. So this is a very area where you'll probably learn a lot about as you go forward in the future. We won't get into electrophoresis, I guess, or we can talk about those another time. We cannot clone organisms. Did you know that you can pay a lot of money to take a DNA sample from your pet, send it to, I think it's South Korea, and they will then clone your pet? Yeah, it's hundreds of thousands of dollars. But basically get a genetic copy of your pet. Yeah, but it's a genetic copy of your. Yeah, some people disagree. Some people say we shouldn't be cloning. So the first cloned mammal was Dolly. Yeah. So yeah, as people have ethical issues sometimes with cloning. We have the technology to clone humans. Okay? Cloning a human is not much different than cloning a sheep. Nobody has done it that we know of, but the materials and tools to do it are not that complicated. That there are many places, many labs in the world where we have the tools and the know-how for people to. People say that they don't think that it's ethical. So if a clone, what is a clone? An exact genetic copy. It's basically, if you were to clone me,
So basically it's a twin.
but it's a twin because if you were to clone me, you would have to take out a cell, take the nucleus out of one of my cells, and that nucleus is all of my DNA. You put that nucleus into an egg cell that's been emptied, zap it with electricity, and it starts to divide. You take that little embryo, implant it into a woman's uterus, and then she becomes pregnant. But she's not pregnant with her child. Genetically, who's she pregnant with? It's my twin. But it's going to be born as a baby. It's not me. It'll be born as a baby. So it's like if I had an identical twin, but they're 45 years younger than me. So here's something even weirder. Technically, using cloning technology, a woman could give birth to her clone. Right? She could have her clone twin and never have any male involved at all. No sperm necessary. She could just clone herself. So basically, you could say with genetic technology, men are obsolete.
I like this idea.
Thanks, Leila.